Hello students, today we will start with a new topic, that is pleiotrophy. Pleiotrophy is the condition when one gene is controlling many characters or traits. Now, we have two examples under this. One is sickle cell anemia and second one is phenylketonuria. So, in both of them, one type of gene that is responsible for their own so-and-so characteristic features are responsible for such types of recessive disorders. Now, first we will see about phenylketonuria. Phenylketonuria is the condition in which there is accumulation of some types of toxic substances in the brain and blood. So first, if we start learning about phenylketonuria, it is also called as autosomal recessive disorder. This example is included in NCERT. So a gene that codes for an enzyme called as phenylalanine hydroxylase. That gene should be present on chromosome 12. When this gene is existed, then it converts the amino acid phenylalanine into another type of amino acid, that is tyrosine. When this enzyme is absent, or when the gene that is responsible to code for this enzyme is absent, then phenylalanine is not converted into tyrosine and its content will be highly accumulated in the body. When this is highly accumulated, then it will be converted into phenylpyruvic acid and that gets accumulated in the brain and it causes damage to the brain. So, when gene that codes for phenylalanine hydroxylase is absent, then phenylalanine is highly accumulated in the body and gets converted into phenylpyruvate, or we also call this one as this pyruvate as pyruvic acid also. Phenylpyruvic acid. This phenylpyruvic acid is considered as a toxic to the brain. It damages brain meninges layers. So, it damages brain meninges. Simply we can write, it damages brain. Now, phenylketonuria is indicated with phenylketonuria. It is indicated in shortcut like this, PKU. You can write this one as capital letter, PKU. Now, this is also called as, considered as autosomal recessive disorder. So what is meant by autosomal recessive disorder? Suppose we have 46 number of chromosomes. That 46 number of chromosomes we are representing as, for suppose if it is a female, we are considering as 22 pairs out of 23 pairs in case of female human as autosomes. Means those are not responsible for the reproductive activity. One pair as a sex chromosome. Now, when there is change in this 22 pairs of autosomes, then, and in that 22 pairs of autosomes also, we know that there are contrasting pairs of alleles forms of genes. For suppose, if a gene that is responsible for some X character, it is because of the dominant one, it always will be having the contrasting another pair of alleles we represent with small alphabets. When there is some change in such type of allele forms of the genes, then that also in case of autosomal recessive autosomal genes or autosomal chromosomes, then we call it as autosomal recessive disorder. There must be change in the autosomes recessive type of allele forms of genes. Sickle cell anemia is an autosomal recessive disorder again. Recessive disorder. Now, this is also considered as a qualitative inheritance. In my first and second videos, I told about variations that are qualitative and quantitative variations, etc., that are not having any effect or change with environmental changes. And I have given detailed explanation about quantitative inheritance and qualitative inheritance. You can please check out that part 2 and 3 for such types of details. Now, sickle cell anemia is the change in shape of RBC that is due to change in the nitrogen base of one type of codon sequence. RBC from. So normally we know that RBC in case of human beings, its shape is biconcave. That is like this. Biconcave. From biconcave to sickle shaped. That sickle shape means it will be like this. Like this. So, RBC has red color pigment that carries oxygen, that is hemoglobin. Now, hemoglobin is biochemically made of 2 alpha chains and 2 beta chains plus 2 beta chains. When we read about quaternary structure of protein, then we also read this one as an example. So, the protein, when it is being synthesized in the body, it takes 4 different stages or structures. Primary structure of the protein, secondary structure of the protein, tertiary structure of the protein, and quaternary structure of the protein. Now, these 2 number of alpha chains and 2 number of beta chains, these 2 are studied under quaternary structure of the protein. This we read in case of biomolecules chapter. Protein. These are considered as the examples for that. Now, in beta chain of hemoglobin, that is, this is the short form to indicate hemoglobin molecule, of hemoglobin at 6th position of codon sequence, there occurs point mutations. There occurs point mutations. So, let's write some types of sequence of amino acids and the codons that are responsible for that. For suppose think that one, 2, 3, 4, 5, 6, and 7. Think that this first codon, some group of 3 number of nitrogen bases, suppose it is coding for glycine amino acid. Amino acids, the names are. This is a phenylalanine. This is leucine. Suppose think this is cysteine. This is arginine. Now, at sixth position, in case of normal hemoglobin, the sixth position should be having glutamic acid. And glutamic acid is coded by some types of codons that are G, A, G or G, A and A. What are these G, A and G? G is indicating guanine nitrogen base. Here A means adenine. So in nitrogen bases, in case of DNA molecule, we have 4 different types of nitrogen bases. Those are adenine, guanine, cytosine and thymine. This is considered to be having 2 rings. So it is purine type, and this one is having only 1 ring, that is pyrimidine we call. Always we know that purines pair with pyrimidines only. But here the codon GAG and GAA, all of them together are coding for one type of amino acid, that is glutamic acid. Suppose the 7th one is arginine. There is no change in any number of amino acid, but only at the 6th position there will be change. Now what happens? At one point of the nitrogen base, there will be some change, and now that change leads to change in the coding of another type of amino acid, that is nothing but valine. When there is some change in the one particular point of the nitrogen base sequence, then it leads to formation of replacing this adenine with uracil. It can be GUG instead of uracil instead of this adenine, or GUA instead of this guanine. But mostly we see that particular amino acid, that is valine, is causing change only at one particular point, that is GUG, and this codes for another type of amino acid, that is valine. So this leads to change of RBC structure from biconcave to sickle shaped RBC. This is about hemoglobin, biochemically how the codon sequence are being changed, so that amino acid that they have to code is also being changed from glutamic acid to valine. So now, if we consider this is the structure of normal hemoglobin beta chain, then what is the sequence of an sickle shaped beta chain sequence of amino acids? So the first one will not change, that will be glycine. Sickle cell anemia, hemoglobin of sickle cell anemia condition. Now, the second one will be phenylalanine. Third one will be leucine. They are not changing. Fourth one will be cysteine. Fifth one will be arginine. So sixth one will be valine. Seventh one will be arginine again. Okay. There is no change in any of the 7th or 5th, 4th, 3rd, 2nd and 1st amino acid sequence or their codon sequences. Normal hemoglobin molecule, it is coded by types of genes, that is, for suppose this A means that type of gene is not coding for any another type of abnormal condition. Normal healthy hemoglobin molecule is coded by this. So if a person is an sickle cell anemia carrier for sickle cell anemia, so his body will be having the allele pairs like at least one type of normal healthy allele is there, that is to protect the organism. Now, affected person has the allele pair as HBS, which is responsible for sickle cell anemia, and HBS. There are very less chances for survival of this person due to lack of oxygen, sufficient oxygen in the body. So these are the 2 examples for sickle cell anemia.